Okay, so we're just going to start by making all of the different plasmid solutions that we need. I've got a rack out of the cupboard there, and I'm just going through now and making each of the tubes, each containing 50 microliters of the substances we need. Then I'm going to make another rack with some blank tubes. So these are the ones we're going to do the mixing in. So we put in the luck first. That's 3.5 microliters into all of the tubes. So we just whiz through that. And then after we've put that 3.5 microliters in, then we'll close that lid and we'll start putting the globin in. Now we'll also close the lids of the tubes that we're not going to put the globin in. That's just a good safety measure. And then we put the 2 microliters of the globin into those. Then we'll put the mutant globin into the last 3 tubes, again closing the lids of the tubes that we don't want to add to and just opening the ones that we do. Then we put the lyre in. Now the lyre goes 2 and 4, remember. So we put 2 microliters into tubes 9 and 12, and then 4 microliters into tubes 10 and 13. And then finally, the pcDNA, 7 microliters goes into the first one, and then variable amounts into the others. I'm not quite sure why they don't add up to exactly the same number, but that's what the protocol says, so who am I to argue? So just finishing up on that with the last additions of the pcDNA, and then we'll have ourselves all the additions done. So close all the lids, and now mix them all. So select the tubes and then press M to mix, and then once they're mixed, deselect again, and then once they're deselected, we might have a look at what the numbers we've got are. So just move that to the side and expand these and show the actual parameters within, and you can see that actually everything looks pretty good. Now we're going to add the fugine, 2 microliters of each, and the instructions say to this one that you have to add it in the middle of the tube, not touching the sides. Actually, funnily enough, that's what happens with all of these additions. When you click on the neck of the pipette tip goes to the inside. So what we'll do now is actually mix by pipetting up and down. So I just set the volume to something much greater, and I just very quickly go up and down, up and down, up and down, and that will mix all the tubes. You can see that they're mixed when we look at their constituents, and they're all ready now to put into the plate. So now we're getting quite a crowded bench, but we'll just keep the pipette set on 12 so that we get as much as we can, and then pipette that into the wells of the plate. Now, this is where I have to do some modification because I need to make it so that these wells are not just a plastic plate, but of course are wells with cells on them. And then when we've done the final one, we should be able to do an incubation and see the results. You can see there's a little bit of residual volume left in there, but that's basically desirable. Okay, so thanks very much for watching that.